start recording. Hopefully this works. There's no catastrophic errors that occur here. But if you accidentally get kicked off, you can jump back in. The pen should stay up throughout this. So basically, this is multiple choice sort of questions here. These should be somewhat similar to what you might find on the test. It might be a little less wordy because I have some character limits I have to deal with. So we'll go through, and the winner gets one free answer. So there's an incentive to do well. So just keep that in mind. You guys ready? We're going to begin. All right. There's also a few kind of jokey questions that there's no points for, but you'll see those as they come up. All right. So which of the following hormones would increase sodium and water? So you basically, so you're logged in, so you just will click the button on what do you think it is, right? So what increases sodium and water and increases potassium excretion? What do you think would be somatostatin, growth hormones, being able to identify which hormones related back to which action within the body? What do we think? And these are good discussion, kind of jumping off points. So we'll kind of go through it, answer it, see how you guys do. I'll kind of figure out where if anyone's kind of going the wrong direction, and then we'll be able to kind of talk about different points about this. Let's not keep the chatter down to somewhat of a minimum. Yeah. See what we think. All right, fantastic. Most people got that correct. So the correct hormone there was aldosterone. You guys remember where the aldosterone is being secreted from? Where would that be? Yeah, so where does aldosterone get released from? The adrenal gland, right? Remember the adrenal cortex there? That's where we're going to be secreting our aldosterone. And remember, this is what we call a mineralic corticoid, right? So it's helping to retain things like water and salt, but at the cost of getting away potassium. So for instance, if you had someone who had a hyperaldosteronism, you expect them to have a high sodium level, but a low potassium, right? Which is otherwise known as? Hypokalemia. Thank you so much. You don't get any points extra for that. Yes, sir.
Which layer was it? Was it the fasciculata or?
That was, I believe, is the outermost area of the cortex there.
Yeah.
Yeah.
There's the fasciculata and the cortex.
Mm-hmm. Yeah. So remember, it was the aldosterone. Then you're going to have the cortisol, and then you end up having the sex hormones. Yeah. And then the insides where what's going to be in the medulla. What was released from there? The adrenal gland. Catecholamines like your epinephrine, norepi, things like that, right? Good. So what does somatostatin do to us or for us?
Somatostatin stops the growth hormone.
Right, so remember, another name for it is growth hormone inhibiting hormone. So that would be incorrect in that case. What does thymopoietin do for us?
Increases white blood cell production.
Perfect, increases white blood cell production, and then growth hormone obviously does what? Grow tissues or organs or bodies. You guys ever seen pictures like bodybuilders who have obviously abused growth hormone? You see their guts are they look like they're almost pregnant because they have all this all their organs develop this hyperplasia that develops because they're using too much growth hormone. Huge muscles, but also big bellies as well. So it's kind of interesting kind of look they have going on. Okay. So everyone kind of comfortable with the format? All right. So up next, all right, I'm in the lead. All right, what is the name of the protagonist in the game Kid Icarus? This is I decided to go Nintendo characters. This is worth no points. It's just for fun. What's his name? So what is the name of the protagonist, this character in Kid Icarus? Yeah, it's no points, so it's just for fun. I had to throw in something jokey just to kind of break it up a little bit. You only get 20 seconds. Actually, wow, I'm surprised most people knew. Yes. Good job. Pit is the correct name. Does anyone know what large gaming event's going on right now? E3, perfect. So it's a good time to talk about it, right? All right. Perfect. Still in the lead. All right. In DNA, which of the following nucleotides would form a complementary hydrogen bonds? What do you think? Be uracil and adenine, adenine and thymine, cytosine and adenine, or cytosine and guanine. This is in DNA. What would you expect to see? What do you think? Keep in mind, on the actual test, there's only one correct answer. Here, though, there may be more than one. Don't get too stressed out. Don't get too stressed. It's to illustrate a point, though. There can be multiple right answers in the world, in medicine. Absolutely. correct. There are two right answers here in this case. So again, this is in DNA, right? So in DNA, there's no uracil. However, if I had said in RNA, what would have been the correct answers? Adenine, uracil, and guanine and cytosine would still be correct, right? So again, those would be correct if I had said in RNA, but we're dealing with DNA in this case. So this would be adenine and thymine, and then cytosine and guanine, right? Cytosine and adenine should never go together. That would be a mismatch. That would usually trigger some sort of DNA sort of DNA polymerase to come in and try to fix it or something like that, right? So we need some sort of fixer to come along and correct that. Perfect. All right. Which of the following organ systems is responsible for movement and support? So again, some of these are going to be kind of basic, kind of memorization sort of questions. Some of these are going to be a little bit more in depth and require to know kind of a couple of steps, right? We're going to have kind of a wide range. Usually in this semester we start out kind of the easiest, the most kind of level 1 sort of questions, and advance from there. But yes, in this case, skeletal muscle, the skeletal system is correct. That would be good for movement and support. In this case, endocrine, not really so much for movement and support. However, certainly useful for lots of other features here. But again, basic facts, basic science stuff you want to kind of know, because this is the building blocks. This is what we're going to kind of develop from further in more sections. Okay. Up next, what's the name of this Nintendo character? These are all randomized, so they sometimes get kind of lumped together. What do you think? Is it Slippy, Fox, Peppy, or Falco? What do we think? A little bit split. Peppy is actually what animal? Peppy's actually what? He's actually a rabbit. Yeah, sorry. So Slippy is the correct answer. He's the toad. Yes. Anyway. It's okay. You guys didn't play enough Star Fox? It's all right. All right. Of course, another one comes up. I only have 4 of these. There's only 4. So who's the main antagonist in the Donkey Kong Country series? This is a little bit more of a lesser-known fact. It's actually King K. Rool. Did he say together it's King Cruel? Anyone know where Bowser's from? Mario. Everyone knew that. Most people knew that. How about King Dedede? Kirby. Yep. Good. And Ridley. It's actually Metroid. Yeah. Okay. Anyway. Okay. The lateral rectus muscle is controlled by cranial nerve. Back to seriousness. So what do you think? 4, 3, 6, or 1? It's the lateral rectus.
How are you going to know if one of this is an option?
Huh? Well, people usually identify themselves at the end. I guess there could be some imposters, but don't you worry. I have ways, okay? Okay, yeah, so in this case, this would be cranial nerve number 6, right? So this is the, what's another name for that? Guys, yes, this is the abductens or abducens, however you pronounce it, yes. How about, what would cranial nerve number 4 do for us? Yeah, superior oblique, right? That's a trochlear nerve, right? So there's that. And then what moves all the other muscles in the eye? 3, perfect. And then 1 we said was what? Yeah, it's our olfactory nerve, right? It's good. Perfect. How about going forward in a car would primarily activate which organ, do we think? Organ of Corti, the semicircular canal, the saccule, or the utricle? So going forward in a car. We're not turning, we're not going up and down, we're just going. Those otoliths are pulling it in one direction, so that way you open up those hair cells and you open up the potassium channels.
Sorry, dude. Maybe next time.
All right, so a little bit of a spread here. Right, so semicircular canals are good for what? Semicircular canals are good for what? Rotational movement, right? So again, that would be for that. So if we were like, say, in a merry-go-round or something, that would be more of the semicircular canals. The organ of Corti is for what? For hearing. The saccules for what? vertical, right? So if I said you're in an elevator and you're going up and down, that would be the sacral-diutrical. However, it's a horizontal acceleration. That would be the kind of the most activated one in this case here, right? All right. All right, so the parasympathetic nervous system uses blank to contract the blank muscle to cause meiosis. So a little bit more in depth here. First question is, what is meiosis? And then what muscles will actually cause meiosis? And then what activates that to cause that meiosis? We know it's the parasympathetic nervous system we're dealing with. Interesting, a little bit of a split there. Okay. So key thing here to note, right? So I say parasympathetic nervous system, you think what neurotransmitter? Always think acetylcholine, right? So you think parasympathetic, you think acetylcholine. Okay, so that's one step you can kind of figure out right there. If I said sympathetic nervous system, I'd be thinking of what? Like epi, norepi, right? So those are the main neurotransmitters I'm thinking of with that, right? So that's how I kind of think of that. Mainly norepi, but you might find some epis kind of elsewhere in the body, but norepi primarily in the sympathetic nervous system. So then the question is, well, what muscles are responsible for causing meiosis? Which meiosis is what? Constriction of the pupil, perfectly. So what muscles are doing that? It's the circular muscles, right? Because we know that the radial muscles do what? They cause dilation, right? They're going to pull that apart essentially when you activate those. So norepinephrine hits the radial muscles to cause what? Mydriasis. On the flip side of this, we're just seeing the parasympathetic nervous system activating or using acetylcholine to activate the circular muscles to cause meiosis in that case. Right? Make sense? And these are good things to know because again, like I use this clinically because I go in and I see someone who's like overdosed on a drug and they have wide, huge pupils. I know, well, one, they either took something that is activating the sympathetic nervous system, or in some cases they're doing the opposite and they actually have an anti-cholinergic or an anti-parasympathetic sort of drug that will do the same thing basically, because that actually will relieve the acetylcholine effect on the circular muscles. They relax and then the pupils will open up essentially, right? So kind of two things you might be actually seeing there, either the agonist effect or kind of an antagonist sort of effect. Okay, we'll get into more detail on that later on in like pharm and things like that, but it comes up clinically more frequently than you might think. Okay. Up next, I'm very disappointed in myself. I'm not even on the top 5 list. Okay, glycolysis takes place in which part of the cell? The Golgi apparatus, the nucleus, the cytoplasm, or the mitochondria? Where is this occurring at? The first question is, what is glycolysis? You have to ask yourself. It's not glycogenolysis. It's a different process, right? All right. Good. So I think most people are along the right track. The question is kind of the specifics here. So again, what is happening in glycolysis? Yep, you're putting in a molecule of glucose and you get what out of it? 2-pyruvate. You get a couple of ATP, but the main thing you're getting is those 2-pyruvate. At that point, that's happening in the cytoplasm. Is that aerobic or anaerobic? It's still anaerobic, right? Because we can use that pyruvate at that point. If we need to convert it over to lactic acid in order to keep glycolysis going, that's possible. We can do that, no problem. However, that's an anaerobic process. If we wanted to then take the pyruvate and then go into what process? Krebs cycle, TCA cycle, right? That occurs in where? That's actually in the mitochondria. And then is that anaerobic or aerobic? That's aerobic, right? So then from those steps on, it's all aerobic from then on. So again, the electron transport chain, the Krebs cycle, that's all happening within the actual mitochondria itself. Okay? Make sense? All right. Again, how many ATP should I get out at the end? The whole process. The very end. So I put in 1 molecule of glucose at the very end of the electron transport chain. I should get 38 total, right? 38 total ATP out of that. You get the baseline. Remember I said you don't have to know all the steps of the Krebs cycle, but you at least know what I'm putting in, what I'm getting out of each of those steps, and that's the main thing right there. That's the whole process. Glucose to ATP, you get 38. All right. What does the glucose eventually turn into at the end of that whole cycle? Carbon dioxide and water, right? Again, everything breaks down to carbon dioxide and water eventually. Okay. Which of the following is in a positive feedback loop? Which would be an example of a positive feedback loop? Oxytocin released during labor, glucagon released during hypoglycemia, release of ADH in response to dehydration, or PTH released in response to hypocalcemia. Perfect, everyone got that one right. Great. So again, so looking back at it, remember a positive feedback loop is doing what? Yeah, it's kind of propagating itself. So what was another example we talked about? Luteinizing hormone during the menstrual cycle? Yep. What was another example? Blood clotting, right? So we talked about during the blood clotting process there. So basically what we're seeing with oxytocin, remember the baby's head is against the cervix, that is stimulating oxytocin release, which then strengthens the uterine contractions, which presses the baby even harder against the cervix, which then releases more oxytocin. So it's building on itself, getting stronger, and then when the baby's out, then that stimulus is gone, oxytocin goes back down essentially, right? In these other cases though, how about glucagon? That is causing the liver to generate new glucose to release into the bloodstream. What's the signal to turn that off? Yeah, the elevated glucose itself is what kind of feeds back and says, hey, the pancreas says, hey, we have enough glucose around here. We don't need any more glucagon, right? And then glucagon is released from which cells in the pancreas? The alpha cells. Remember, insulin comes from the beta cells. But again, in this case, glucagon, when the pancreas detects there's enough glucose around it, it can shut off glucagon release. Okay, that's the primary thing that's going to be shutting off. It gets more complicated as we'll see, but that's the main thing. How about release of ADH? Again, what does ADH stand for? Antidiuretic hormone. What's another name for it? Yeah, vasopressin is the other name for that. So again, this is a response to dehydration. So in this case, ADH does what? So you hold on to sodium and some water. Okay, what else does it do? Cause some vasoconstriction. What else does it do? It's just stimulating centrally. Thirst response, right? So again, you're getting more thirsty. You're going to be trying to take in some water. That's the main thing it's going to be doing there. So again, once the body, and what is the hypothalamus detecting when it's releasing ADH?
Low blood pressure, low blood volume.
Not the volume of the blood or the pressure necessary. The more important thing is going to be what? Yeah, the osmolality of the blood. So again, when it detects the blood's too concentrated, that's usually what's stimulating ADH release. Because again, that wants to hold on to more water, and it's going to help to dilute out, basically dilute the blood essentially. And then when you get to a point where the blood is now at a good osmolality, that will then decrease the release of ADH, right? So that's a negative feedback loop in play there. Okay. And then parathyroid hormone or PTH is coming from where? Perfect. So it's detecting that there's low calcium, and then what does that stimulate? So yeah, so release parathyroid hormone, then it does what?
GI, kidney.
Yeah, so the bone, what does it do in the bone? Stimulates osteoclast, good to release more bone from our main storage site. Well, it says GI tract, what does it do there? Increases absorption, good, and then in the kidneys. It actually increases reabsorption of the calcium, right? So again, you filter a lot of stuff in the kidneys, but then you reabsorb a lot of it. This stimulates reabsorption of calcium, so that way you don't lose it out in the kidneys, right, in the urine. Again, that is a negative feedback loop because once the calcium raises up to normal level, what does it do to the parathyroid hormone release? Shuts it down, right? So again, those are all examples of negative feedback loops. So it's good to kind of understand kind of the process here and what is going to be the brakes on the system. Because again, if I had a case where I had maybe some kind of parathyroid, maybe a PTH secreting tumor or something, and I was releasing too much parathyroid hormone, that signal never gets shut off, then you can see things like the bone starts to break down too much. You can end up seeing osteoporosis develop, all kinds of problems if that negative feedback loop is interrupted. Yes, sir?
I was looking over it last night, and it's the gonadotropic inhibiting hormone,
Mm-hmm.
hormone. Doesn't estrogen also cause it to stop releasing gonadotropic releasing hormone?
Yeah, so that's a good point. Someone mentioned that a few minutes ago as well. So basically, for some of the hormones we talked about, there's kind of the agonist, right? So in the case of like gonadotropin releasing hormone, there's also kind of an inhibitor as well, right? So in and of itself, something like estrogen or testosterone can feed back and shut down further release of gonadotropin releasing hormone. However, it may also stimulate something like the release of the inhibitor, right? So it just depends on the situation.
Is that considered negative feedback loop still?
If the end product is what's feeding back and trying to shut down the system, then I would call that a negative feedback loop, essentially. Yep. All right, perfect. All right. Movement of glucose into a cell through the GLUT3 transporter is an example of what? Osmosis, simple diffusion, active transport, or facilitated diffusion. I like randomizing this. It's good. You have to kind of jump from like, you know, topic to topic, just like you do on the test, and kind of get your mind used to kind of switching gears pretty quickly there. Perfect. Okay, so let's break this down. So it's not osmosis, because what is osmosis? Mainly just movement of water, right, across semi-permeable membrane. So that's not going to be the case here. Simple diffusion is just what? Yeah, so if they have like a really lipophilic molecule and that can cross the membrane by itself, that would be a case of simple diffusion. And again, that's going from areas of high concentration, areas of low concentration, right? It's flowing along its gradient. With active transport, though, obviously that's going to require something like ATP in order to function. Also, what is it normally doing as far as pumping things from one area to another? Usually against the gradient. Usually you're going to find that, because that's why it requires energy. So again, the body tries to be efficient when it can, but in this case you have to use energy to go against the concentration gradient. Again, a good example is the sodium potassium ATPase pump, right? Using ATP to pump things against the gradient. Usually sodium outside of a cell, potassium into a cell. And in the last case here would be facilitated diffusion. This is just using a transporter, but it doesn't. It's an active. It's a passive process, right? It just needs that transporter though to interact with the glucose and says, okay, this is glucose. Let's open up and allow them to go in, right? So once insulin comes along, triggers the cell, say, hey, we need to go ahead and export these glucose transporters, moves up to the cell surface, and then glucose again going along the concentration gradient, because usually it's higher in the bloodstream than it is within the cells. Go along, hit that transporter. It's facilitated, but it's still not an active process. So you kind of see the difference there. Okay, so that's an example of that. All right. Which of the following molecules is the starting point of the Krebs cycle? It's glucose, acetyl-CoA, pyruvic acid, or lactic acid. So, what are we putting into each of these steps, and what are we getting out of it are good things to know. Good. So this is the key point to note here, right? So again, it's not glucose, right? Because glucose is starting off what? That's the thing that kicks off glycolysis, right? So that would not be the correct answer. Lactic acid is also not correct. When do we produce lactic acid? Yeah, during anaerobic sort of conditions, right? Then we use that pyruvate and convert it over into lactic acid. Acetyl-CoA, though, remember that pyruvate that we generate during glycolysis has to be converted over to acetyl-CoA, and then that's what goes into the Krebs cycle, right? On the other hand, pyruvic acid itself is just not really doing anything. It can either get converted to lactic acid or it goes into and turn into acetyl-CoA. See the difference there? So again, you have to have the intermediary step to occur, so that way you have acetyl-CoA and that enters the Krebs cycle. Okay, so little details there you want to kind of key in on. Okay. Which of the following molecules will be considered polar? I should have put like bear up there as a answer. So CO2, O2, CH4, or H2O? Yeah, most people got that one right. So again, remember if you go back to how these molecules are kind of arranging themselves, remember that H2O, remember how water kind of forms a Mickey Mouse sort of conformation there, where again the hydrogen ions are kind of closer to one another, kind of attracted to one another. And in that polar area where we have an area of higher positive charge on one end, and an area of more negative charge on the other end, that's super important. Why do we care about that? Solubility, right? Because again, if stuff doesn't solubilize in our blood, which is a lot of water, guess what? We're going to have problems, right? So we need things to be soluble in there. So knowing that that's a polar molecule is a good thing to know, because that tells us if things dissolve in it or not. On the other hand, most of these other molecules have basically kind of symmetrical sort of shapes, and they tend to be less polar, they tend to be non-polar, they tend to be immiscible, or not be able to mix in water necessarily, right? So things like methane, which is that CH4, again, has a very kind of stable sort of shape, all it's sharing its charges kind of equally around itself. Similar for O2, there's just two oxygen molecules, very kind of symmetrical shape there, and then CO2, again, very similar to that. So main point being is that you want things to be to dissolve in water. They need to have some degree of polarness to it. That's why we see things like salt dissolve into it very easily, because when it dissociates into sodium and chloride, it has positive charges. It's able to orient itself with the water very easily and become soluble, essentially, right? Make sense? Okay. All right, so if calcium has 20 protons, 20 neutrons, and 20 electrons, what is its atomic mass? 40, 10, 20, or 60? All right, most people got that correct. So how do you get that number? Protons plus neutrons. Perfect, right? Because electrons weigh what? Almost nothing, right? For our purposes, nothing. Right, so again, that does not get included in there. If I said what was the atomic number of calcium, you would say 20, because that's based on the actual protons it has there. Good. So again, kind of keep those distinctions in mind there. So 20 would not be correct. 60 would not be correct, because again, the electrons, for its mass purposes, is negligible. It's nothing, basically. Okay. So again, you may have to do some very basic math on the test. There's a question like this. Again, you're not going to need a calculator, so don't worry about that. Maybe your scratch paper if you need to, or your fingers and toes. You can take your shoes off if you like. Okay. Which of the following RNA molecules is used to transfer genetic information to the ribosomes? MicroRNA, transfer RNA, messenger RNA, or pre-messenger RNA? All right. Okay. So that process where we're getting information from the DNA and turning it into RNA, like a complementary strand of RNA, what do you call that? Transcription, right? Yeah, transcription, right? So that's how we form our initial set of RNA. Now, again, that itself is going to be end up needing to be spliced and kind of modified in order to, before it gets transmitted over into to the ribosomes, right? So again, you're going to start off, and that's where things like microRNA can be useful to kind of help to guide how you're going to be chopping and splicing that molecule RNA, right? Once it goes from RNA, that is considered, once it's ready to go to the ribosome, you call that what? That's messenger RNA, right? Because it's taking all the information from the nucleus, and as a messenger, it's carrying it over to the ribosomes, where then it can start the process of translation, right? So again, translation occurs once the messenger RNA gets over and interacts with the ribosomes. Now, what brings in the amino acids that reads those codons? That's the tRNA. That's the transfer RNA there. So it's important to kind of keep those distinct. And again, you may say, like, why the heck do we care about this stuff? Why do we care about ribosomes and RNA? This becomes important when you're talking about things like cancer drugs, when you're talking about antibiotics, when you're talking about all sorts of antiviral drugs. At least for my purposes, this comes up very frequently in the pharm class. So kind of having a good understanding of how these processes are occurring is a good thing to know. All right. Which of the following solutions would cause cells to shrink and crenate? 0.22% sodium chloride, lactated ringers, 3% sodium chloride, or sterile water. It goes back to tonicity solution. Things are isotonic, hypotonic, or hypertonic to our blood cells, our cells. What do you think? This is assuming it's a human cell at a normal osmolality in the body. All right, the correct answer would be 3% sodium chloride. Now, going back to looking at tonicity, so again, there's hypotonicity, which would be the concentration of solute in the fluid to be what compared to the cell we're looking at. Hypotonic should be a lower concentration. It should be a more dilute solution, right? If it's isotonic, how's the concentrations of solute compare? They're equal at that point, right? So again, in that case, you should not see any fluid shift. If it's hypertonic, the solution will be more concentrated, right, than what's in the cell, okay? And that's how we're going to be shifting these fluids around. So in the case of like lactated ringers, that's considered to be what? That's isotonic, right? So that's why it's a really good fluid to give for like patients getting surgery. Give them isotonic lactated ringers, you shouldn't see any fluid shift whatsoever, right? What would be another example of an isotonic fluid? Normal saline, which is what percentage? 0.9. That's a good number to remember. For normal saline, 0.9% sodium chloride. So if I were to give a hypotonic solution, say for instance something less than 0.9% sodium chloride, like say 0.22%, which would be quarter normal saline, right? Because again, if I did half of normal saline, it's 0.45, and half of that is roughly 0.22, so that's quarter normal saline. If you ever hear that, that's what we're referring to. Or sterile water, which has no solute whatsoever, that's going to do what to water within that cell? It's going to go into the cell, right? Because again, the water wants to go try to make the concentration on either side of that similar, so it's going to cause fluid to go into those cells, and that will cause the cell to do what? Swell, and then eventually lyse if it's too much, right? Which is why you never, ever, ever, ever want to give sterile water via IV. Actually, we don't even let that out of the pharmacy, because if you were to inadvertently give that to a patient, you can kill them very easily, right? Because again, you never want to dilute out the blood that much. It cause all kinds of problems. You don't want to do that. So isotonic fluids, we said, would cause no shift whatsoever, so the cell would stay exactly the same. However, if I were to give a hypertonic solution, like 3% sodium chloride, which is over 3 times as concentrated as normal saline, that should cause what? Water to shift out of the cell, right? That would cause the shrinking to actually occur there. And again, we do that. If you have a head trauma patient who got cerebral edema and cerebral swelling, we will give 3% saline to draw water out of the CNS and relieve that pressure, right? So again, we do that clinically. If you work in a trauma center, you'll do that with some regularity, right? That makes sense? All right. During repolarization in an action potential, decrease in membrane voltage would occur due to what flow? Would it be chloride efflux, sodium influx, calcium influx, or potassium efflux, right? So this is the repolarization period right here. What do we think? Now this is in general, right? If we're talking about, say, like a hair cell within the organ of Corti, or the, say, like the semicircular canals, this will be a little different. We'll talk about that in a second. But primarily, what is causing that repolarization to occur? It's actually potassium efflux here, right? So remember, what is causing that initial rise in the electric potential there? What's causing this increase? That's sodium influx, right? Because remember I mentioned our cells are kind of salt-wrapped bananas, essentially, right? So there's a higher potassium concentration in the cell, higher sodium on the outside of the cell. When you have depolarization occur, sodium channels open up and sodium flows in. That's what causes that rapid rise in electric potential, right? Now, how do we reset that? How do we get back down to baseline? Well, you got to open up potassium channels, and that will allow potassium to flow out of the cell, and that's where you see this big drop here in the potential. And in fact, it actually goes to this hyperpolarization, where it actually goes below what the actual resting membrane potential normally would be. Okay, so that's all potassium. Once those close up, what kind of resets the resting membrane potential, though? Sodium, potassium, ATPase pump. Remember, that's the main thing that's going to help us to maintain that resting membrane potential. Okay, so those important concepts. When we get into cardio, this is going to become super, super important. When we get to neuro, also very important there as well. This is going to come up again and again. Okay, now when I say refractory period, what does that mean? Yeah, basically means it's a period of time where it doesn't matter how many more action potentials come in to try to hammer that cell, it's not going to kick off another action potential. It's still trying to reset. It's like, hey, I'm too tired here. I can't do anything right now. Let me go ahead and get back to resting membrane potential. And then once you're out of the refractory period, now you can have another action potential occur, right? So that's important, because when you modify that refractory period, if you make it shorter or longer, that's where you can see things like seizures develop. That's where you can see things like arrhythmias happen. So refractory periods are very important to understand as well. Okay? Are with me on that one? All right. Up next, having a shortened eye that focuses images behind the retina can lead to which vision disorder? Myopia, hyperopia, astigmatism, or emmetropia? There's a shortened eye, images are focusing behind the retina. Again, I have to go and remind myself every single year of this, because I don't deal with the eyes very frequently, so I got to re-educate myself. Interesting. Okay. So, hyperopia, otherwise known as farsightedness. That's usually a case where you're going to have the eyeball is too short, and you're actually focusing images behind the retina. Okay. So the question is, how do I go and try to correct that? How do I get the image to try to refract more, to try to refract back on the retina? Do I want a convex or do I want a concave sort of lens? You want a convex lens, right? You want to bend that light more so that way it kind of bends it closer, right? It's going to try to bring it closer and focus it right in the back of the retina, okay? Now, if I had myopia, otherwise known as nearsightedness, it's usually when the eyeball's too long and then the image is focused where? It's in front of the retina, right? Because again, the eyeball's too long at that point. So how would I fix that? Yeah, if I use a concave sort of lens, that's going to refract that light out. It's going to make it go farther and actually hit the back of the retina, right? It's going to hit that fovea, where you know a lot of those, you get the sharpest image essentially right there in the back of the retina, okay? How about astigmatism?
They go everywhere.
Yeah, it kind of goes everywhere. It's an asymmetric sort of lens there, right? So again, it's, the light's concentrating or is focusing on several different points, right? So that's where he needs kind of a cylindrical sort of lens or something like that to try to bring it all into focus, essentially, right? So that's kind of the big difference with there. Hyperopia is what? That's just normal vision, right? Now, if I said presbyopia, what would that be? Old age, you know, vision getting poor as get older, the lens gets less flexible, things like that, right? So that's kind of the difference there. Okay, everyone, that makes sense to everyone? Okay. All right. Overactive parafollicular cells in the thyroid gland could lead to increased levels of calcitonin, iodine, thyroxine, or PTH. What do we think? The one straggler. What do we think? All right. Calcitonin would be correct here. So, remember, the follicles are where we're actually producing thyroxine. And remember, there's T4 and T3. Which one's thyroxine? T4, right? But we know that the more active form is T3, absolutely, right? So again, those are going to be the follicles of the thyroid gland, but the parafollicular cells, those are responsible for releasing calcitonin, okay? Calcitonin does what for us? Decreases calcium levels, right? So you can think of that as the antagonist to PTH, right? Because PTH detects low levels of calcium and tries to increase that. Calcitonin does the opposite. It's going to be detecting high levels of calcium and try to lower those. How would it do that? Okay, so, well, more likely it's going to inhibit osteoclast, but yeah, you'll have some osteoblast stimulation. What else can it do? Increase excretion where? In the kidneys, good, and then increase absorption or decrease absorption from the GI tract, right? So again, it does the opposite of what PTH is going to do, and that's from those parafollicular cells in the thyroid gland. Iodine wouldn't be correct, but iodine is important because what do we use that for? Yeah, it's a precursor to T4 and T3. If you're missing that, you're not going to be able to produce any thyroid gland, then you're developing what? Yeah, hypothyroidism and then potentially a goiter, right? Because again, if you don't inhibit that negative feedback loop, you have all that TSH overstimulating your thyroid gland, ends up getting enlarged, right? Now remember, you need enough iodine, but you get too much iodine, that can actually inhibit thyroid production as well. So that's also another big problem with having too much iodine around. Not usually a clinical issue for most people, but it is a potential concern. Okay. Go dill pickle. Okay, during heavy menses, which hormone would be stimulated to replace lost red blood cells? We got aldosterone, erythropoietin, cortisol, or oxytocin. Perfect. Yeah, so erythropoietin would be correct. So that's going to be stimulating red blood cell production. Normally where? The marrow. What type of bones though? Flat bones like the sternum. Also get like in the spinal column, things like that, right? So those are the main places you have red blood cell production. Where does erythropoietin come from? The kidneys, right? So again, that's a big problem you run into with patients with chronic kidney disease. Is they typically have decreased erythropoietin levels. You have decreased erythropoietin, you have decreased red blood cells, and you develop anemia. What type of anemia? No, not hemolytic. Huh? Not aplastic. No, basically, so their anemia is basically because don't have any stimulus to actually produce it. So even if they have all the precursors, they have enough like B12 and they have enough, iron and all that good stuff, they just don't get the stimulus to actually produce it. So actually, in that case, it'd be a normochromic, normocytic sort of anemia, right? You just have a decreased number of them. You're not increasing lysis of them or anything like that. However, if you have something like decreased iron, decreased other cofactors that are needed for that, that's when you run into some of the other types of anemia, right? But again, in this case, it's just the erythropoietin's a problem. Okay. So in chronic kidney patients, that's a big issue. You run into them quite frequently. The other problem is when I put them on dialysis, remember that's that artificial kidney we'll put them on. Again, whenever you're circulating blood out of the patient, you end up losing some of that in the circuit. So not only are they going in, getting frequent lab draws, they're having blood loss during the dialysis, and now they have decreased erythropoietin. Anemia is a big problem for them, right? And what's maybe a side effect of having anemia? Fatigue's one of the biggest things, right? So again, fatigue, they just have no energy, and then it's not good for them. So that's why we'll sometimes give replacements. But anytime you're giving something to try to stimulate erythropoietin, you got to give iron with it. That's another big key point we'll try to mention later on. They have to have iron. Don't have iron, doesn't matter how much erythropoietin I have, it's not going to do me much good. Because iron goes into what? Hemoglobin. Yeah, so we'll talk about hemoglobin later. That'll be important in the kind of some upcoming sections. Thank you. All right. What is the end product of meiosis 1? There's meiosis 1 and meiosis 2. What do we have? 2 haploid cells, 2 diploid cells, 4 haploid cells, or 4 diploid cells? All right, there's a little bit of division here amongst, get it, division? Anyway, okay, so there's meiosis 1 and meiosis 2. Meiosis is trying to do what? Produce our sex cells, right? Either sperm or eggs. And so in the case when you're actually having the initial division there, the end of meiosis 1, what do we have? We have 2 cells, right? And the initial mother or father cell is going to divide, and then we're left with 2 cells, right? So again, you can already get rid of the 4 diploid, 4 haploid cells. The question is are they diploid or haploid? What's the difference between the two? Yeah, so haploid means they only have half of the necessary number of chromosomes. Now in this case, during meiosis 1, they have an extra copy of it, right? But they only have half of what they actually need for that process to occur during meiosis 2. In the case of mitosis, you would have what at the end of that? You'd have 2 diploid cells, right? Because at that point, they still have each copy of each chromosome, so they're good to go. In this case, at the end of meiosis 1, there's 2 haploid cells. They just happen to double up on the chromosomes because that happened during the S phase there. And then under meiosis 2, that's when they're going to divvy up into what? 4 haploid cells at that point, right? So again, 4 haploid cells should be the end product of meiosis 2. Make sense? Okay. All right. Up next, what's the name of this Nintendo character? I don't remember what you can put down there. Anyone know his catchphrase? I'm gonna win. Slightly racist? A little bit. Okay, yes, that is a Waluigi. Wario is the large yellow fellow, and then obviously we know Mario, and then Toad's the little guy with the mushroom head, right? All right, so Waluigi is the correct answer. All right. Sweet taste is mediated through which type of receptor? Sodium channels, tyrosine kinase coupled receptors, H plus channels, or G protein coupled receptors? Good. I know we just covered that today, however. Still going to be on the test, so I cover it here. So again, sodium channels would be good for what? Salty perception, right? So again, that would be for that. How about for the H plus channels? That was going to be for sour. G-coupled are going to be for what? Sweet, bitter, umami, right? So again, you get those three through G-coupled receptors. Now remember that the bitter versus the sweet and the umami tend to be, they'll have different downstream effects, right? Depending on what sort of secondary messengers are going to hit. In this case here, though, sweet is still mediated through a G protein receptor. This is kind of a red herring. There'd be no tyrosine kinase coupled receptors responsible for taste. Okay, make sense? Okay. Again, if I said smell, what would that be mediated through? Smells all G proteins, right? We just have a ton of different types of G proteins that respond to different sort of odorant sort of molecules. Okay. All right, which enzyme catalyzes cGMP or cyclic GMP and causes sodium channels to close when light affects rhodopsin? So going back to vision, what's happening here? The adenyl cyclase, RNA polymerase, phosphodiesterase, or lactate dehydrogenase? This is catalyzing cGMP. Talked about some specific examples of this occurring in some case of modified vision due to some medications. All right. So adenyl cyclase does what?
That's to make cyclic AMP.
Yeah, so that converts ATP into cyclic AMP. In this case, we're talking about catalyzing cGMP. What does that mean? We're breaking it down essentially, right? So again, we're catabolizing it, right? So I'm catalyzing that. So that was breaking it down to cGMP. So in the case of the eye, what's interesting is with the normal cells, and you're triggering an action potential, normally you're trying to let sodium in to depolarize the cell. In this case, normally those sodium channels are open when they're in a low light sort of setting. But however, when light comes in and starts to catalyze rhodopsin, that's actually going to cause cyclic GMP to be broken down, usually due to phosphodiesterase, and that actually will close those sodium channels, and now you've hyperpolarized the cell, right? So the rods and the cones are a little bit different than most other cells. And by doing that, that's what triggers off the downstream ganglion stimulation. It says, hey, there's light here, right? They're detecting a vision. What does RNA polymerase do for us? Yeah, so it produces RNA from DNA strand. Good. And how about lactate dehydrogenase? Yeah, so this is basically going to be helping us convert things like pyruvic acid over to lactate. It can be useful for things like anaerobic metabolism and whatnot, right? And again, you're thinking about like anaerobic metabolism. Say you produce lactate. What happens to that lactate? Does it just stay there in the cell? Yeah, it gets transported to the liver, and then what happens from there? So some of it can be turned into pyruvic acid, but eventually it's going to turn back into glucose, right? So remember, it's not as an efficient process, but in the liver we can convert lactic acid back to glucose and then put that back into the bloodstream, right? And remember what that cycle is called? The Cori Cycle, right? So Cori Cycle is helpful for helping us to recycle that lactic acid. Again, it's not as efficient, but it's still able to utilize lactic acid for some purpose. Okay, good. All right. Carbonic anhydrase is responsible for catalyzing which reaction? Does it put sodium chloride together? Does it break down bicarbonate into CO2 and H2O? Take CO2 and H2O, convert it to bicarbonate. What is it doing? So remember, this is really helpful for our buffering system within the bloodstream, right? This helps us to regulate pH. And we'll go into this on more detail, especially when we get into the heme section a little bit later on. So there's actually two right answers here. So carbonic anhydrase is really important because it helps us to make sure we can buffer the blood appropriately, right? So normally, if you have bicarbonate floating around the blood, this is what helps us to buffer those extra hydrogen ions that get produced in a more kind of acidic medium. So as you have more hydrogen ions in the solution, as the concentration goes up, this will combine with that bicarbonate to form this carbonic acid, right? Now, obviously, sodium chloride would not be correct here, but the thing that carbonic anhydrase does for us is either convert CO2 and water over to bicarbonate, or carbonic acid, or it does the flip, and it can actually convert the carbonic acid into CO2 and water, right? And so by doing that, it can help to either produce new carbon dioxide or help to convert it back over. And then that way, the CO2 can be transported to places like the lungs, where it can be breathed off, wherever it needs to do. Okay, so again, it's an important enzyme. We'll talk about that again in the kidneys. Very important for the kidneys. This is very important. Anyone familiar with the term like altitude sickness? This is an important process for altitude sickness, as we'll see. We'll talk about that later on as well. So again, this enzyme will come up again and again, but especially in the heme section. So kind of keep that in the back of your mind. All right. Removal of the adrenal glands would cause which of the following? Cause high cortisol levels, low CRH levels, high ACTH levels, or low somatostatin levels. So it's going back and looking at the different steps of that HPA axis, right? All right, so let's look at this. So high cortisol levels, why is this incorrect? I took out the adrenal glands, so there's nothing left to produce the cortisol. So that would not be correct in that case, right? How about low CRH levels? Where does CRH get released from? The hypothalamus. Good. So in this case, if I'm not inhibiting that negative feedback loop, it should be what? It should be high in that case, right? Because it says, hey, there's not enough cortisol around. We need to stimulate this to make more. More CRH would be released in that case. So high CRH levels would be expected. Low somatostatin, we know that's not going to be correct because somatostatin is, it has to do with inhibiting growth hormones. So that would not be correct. But high ACTH levels would be correct, right? Because again, this gets released from what? Anterior pituitary, and that is going to be trying to stimulate the adrenal glands, which are no longer there. No negative feedback loop means it's just going to try to increase that release, right? So I could have said something similar. I could have said, if I removed the thyroid glands, you could have said high TSH levels would be high in that point, right? So again, all of these are very similar in how they're acting. It's just a matter of knowing kind of which hormones are going with which system, essentially, okay? All right. All right, Adam Wood coming back. We got like 2 more questions, so. Which cycle allows for the recycling of lactic acid in the liver? Wish I had not given that away already. Free point for everyone. Unless you have very short-term memory issues, and then no point for you. Everyone's like, wait a second, what did he say? Yes, that would be the Cori cycle. So we mentioned the Krebs cycle, otherwise known as citric acid cycle, right? That's going to be located in the mitochondria. It's helping us to get ready for the electron transport chain, so that would not be correct. And glycolysis is what? Breakdown of glucose into 2 pyruvic acid molecules, right? 2 pyruvates. Good. The Cori cycle, that's what helps us to recycle the lactic acid within the liver, convert it back over to glucose typically. Okay. All right. In the last question, which of the following is a byproduct of oxidative phosphorylation? So O2, H2O, CO2, or H2O2? That's another name for that electron transport chain, oxidative phosphorylation. Two correct answers once again, yes. The end product of the whole entire process of converting glucose into ATP should be byproducts, or CO2 and water, right? Again, that's during oxidative phosphorylation, requires oxygen, but should be producing carbon dioxide and water. What's H2O2? Hydrogen peroxide. What's that used for in the body?
Is it a peroxisome breaks it down?
Yeah, you can see in the peroxisomes, like in lysosomes, you can utilize that to help break down things. It's a very reactive sort of species that can be used to break down proteins and things like that. And then O2 obviously is going to be one of the inputs into the system that helps it to generate ATP, right? All right. So, the podium, we have number 1, Adam Wood. Yes. Good job. Fantastic. There's no collusion here. No collusion, right? Dill Pickle, is that you? All right, perfect. Eugene? Eugene? Good job. Fantastic. Are you ready for your free answer?
Yes.
C. All right. So, any questions for the test? Everyone feel like that was useful? Everyone feel like they have a better idea of what the test might be like? They're similar questions, not going to be the exact same questions. However, this is my thought process, this is how I come up with questions. This is what, similar things might be on the test essentially. So I'm going to go ahead and post this video. So I'll post that up so you can go through and listen to it again if you like. I will also post a link to the actual quiz so you can go in and take it yourself if you like. So you can go back through it and kind of quiz yourself before the test if you want. Okay, any questions I can answer beforehand? Okay, I will be available. I try to answer emails and questions as late as possible before the test, but however, if you email me at 2 a.m. on Thursday, probably not going to be answering that, right? Or Friday morning, I guess I should say. So again, keep your expectations within range there. All right, no, I will see you guys next time.